Hi guys, I'm Mujtaba Ahmad and I am back with the 9th lecture in this video series, and today's topic is going to be on the idea and on the concept of how chemistry and biology interact together. In fact, we are going to talk about the very fundamental ideas in the field of biochemistry. But what you see on your screens is an athlete that is seen to be consuming banana. And we often see, and it's a common sight to see these athletes or long runners or gym enthusiasts having consuming these fruits such as bananas or other fruits and foods which are a rich source of carbohydrates. And that's because carbohydrates are found to provide with instant energy, fuel the body with the necessary stamina, strength and stamina for long running or for strenuous physical exercise, and that's why it's a popular choice amongst these athletes or fitness enthusiasts. So this is the topic where we are going to talk somewhat similar to what I've talked already about. So it will be about the biological molecules, the carbohydrates, proteins, fats and nucleic acids, which are really the foundational ideas in the field of biochemistry. So it's really important and in fact an interesting topic to learn. So the learning outcomes include the chemical elements that make up these biological molecules which I have just named, and then we are going to state only that these large molecules are made from smaller molecules. So I am going to discuss the idea of monomers as well, and then polymers as well. Limited to starch, cellulose, glycogen from glucose, and the proteins are composed from the building blocks which are amino acids, and lipids from fatty acids and glycerol molecule, and then DNA is composed from the nucleotides. Then we are going to be quickly describing the food essential tests for these food molecules, these biological molecules, which include starch, which would be carried with an iodine solution, then Benedict solution test for glucose and maltose, essentially the reducing sugar test, it's also known as that by that name. And then protein are tested with the Biuret reagent, and ethanol emulsion test for lipids or fats. So macromolecule, monomer, just a minute. So yes, macromolecule are the biological macromolecules are large organic molecules which are such as the carbohydrates, lipids, proteins and nucleic acids. Then we are going to discuss this the idea of monomer, which are the simpler molecules which are essentially the building blocks for larger molecules, which are polymers. For example, an amino acid, which I just talked about, is the building block for a protein. And then polymer are large molecules which are comprised of repeating subunits of monomers. So for example, we are going to see carbohydrates are large molecules. They are polymers, and they are composed of repeating subunits of glucose. Okay, so poly means many, mono means one. Okay, so monomers are the building blocks. They are one molecules, and the repeating subunits, when they are repeating when they are in a repeating manner or repeating fashion, they are a large molecule is specifically termed as a polymer in science. So this is what I just wanted to make clear before going on to the next ideas. Carbohydrates, you need to be able to understand the formula or define this term. Carbohydrates, they are represented by CH2 whole brackets NN. So in other words, the ratio of hydrogen in this molecule is 1 to 2 to 1. So hydrogen molecules are twice as compared to carbon and oxygen in a carbohydrate molecule. And the simplest one is glucose, which is C6H12O6. This is the simplest carbohydrate that you will know. And carbohydrates can be classified into three subtypes. So one is monosaccharide, disaccharide and polysaccharides. Saccharides are sugars, okay. Mono means one, di means two, and poly means many. So monosaccharides are comprising of one unit, one like glucose. Then disaccharide are the sugars which itself are composed from two simpler sugars, for example sucrose, which is comprised of fructose and glucose, which is a common one found on a table sugar. And then polysaccharides, starch for example, they are composed of repeating monomers, okay. Starch is the stored form of sugars in plants and is made up of amylose and amylopectin. So these are two major repeating subunits. They are repeatedly found in the structure of starch, and that's why it gives rise to this polymer structure. Glycogen is the storage form of glucose in humans and other vertebrates and is made up of monomers of glucose. So you need to understand that it's made up of simple repeating units of glucose. Then cellulose is one of the most abundant natural biopolymers. The cell walls of plants are mostly made up of cellulose, which provides structural support. So it makes sense that it is the most commonly found sugar all around the globe, because it is found, or it is the fundamental unit of cell walls. So that's why plants are everywhere. We remember plants are everywhere. So cellulose is in fact everywhere, because cellulose is the fundamental unit that's responsible for the composition of a cell wall. And then wood and paper are mostly cellulosic in nature. Cellulose is made up of glucose monomers. So you have to understand and state that the repeating subunits of these polymers are glucose monomers that are linked by bonds between particular carbon atoms in the glucose molecule. So you do not need to understand the details, but you need to be able to state that these all three are the monomers are composed from the monomers of glucose, which is the simplest sugar. Then lipids. Lipids, they include a diverse group of compounds. There are in fact many other elements aside from carbon, hydrogen and oxygen that you will find to be categorized as lipids. For example, sphingolipids, for example phospholipids, they are a crucial part of cell membrane, and they also include phosphate ions. Lipids are generally hydrophobic. Phobic means it's from phobia, fear, okay. Hydrophobic means water faring. They are not insoluble in water, and they give cloudy appearance when they are dissolved in water, because they won't dissolve, and they are non-polar molecules. Water is a highly charged molecule. In my last class I also told you that it comprises of partial positive and partial negative charges. So that's why it's highly polar molecule. And on the other hand, lipids are non-polar. They are not charged because there is no dipole or other force available in amongst its bonds. And this is because they are hydrocarbons that include only non-polar carbon or carbon hydrogen bonds. So that's what I just told you. Cells store energy for long-term use in the form of lipids called fats. So lipids are composed of carbon, hydrogen and oxygen atoms, and in some cases phosphorus, nitrogen, sulfur and other elements. That's what I just told you. A fat molecule such as triglyceride consists of two main components, glycerol and fatty acids. This is what you need to understand. The fats are composed of glycerol and fatty acids. Like starch was composed of glucose, repeating units of glucose. You must understand that fats are comprising of glycerol. You must be able to understand and write is glycerol plus fatty acids. So this is the general subunit or subunits that comprise a lipid molecule. Now the one of the most important ones, proteins, which are found almost everywhere, are the most abundant organic molecule in living systems and have most diverse range of functions of all macromolecules. So this is very true, because from the beginning of life, from the initiation of transcription and translation and DNA replication, which are all the stages where the hereditary material is copied and passed on to the next generation. Proteins are really there. They are like the workhorses in a cell, in a body, and they also carry or catalyze a multitude of metabolic functions in our body. So proteins may be structural, regulatory, contractile, or protective. They may serve in transport, storage, or membranes, or they may be toxins or enzymes. So virtually they are performing multitude, or really they are very versatile molecules, biological molecules, and they are absolutely important in carrying various bodily functions. You must have known by now that they are structural, they have structural roles, regulatory roles, contractile roles, protective roles. They are forming as catalytic agents which speed up the rate of chemical reaction, and we are going to see the enzymes, which are essentially proteins in nature, in the very next lecture. So there are about 20 amino acids, which are the so amino acids are the building blocks of proteins, and they are composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. So there are many different amino acids that link together to give a protein its structure, and therefore every protein is specialized in carrying out their own specialized function, own specific function. The functions of proteins are very diverse because there are 20 different chemically distinct amino acids that form long chains, and the amino acids can be in any order. So this is very important because this amount is enough to provide or to impart variety, or you can say difference in various protein structure, because essentially it is the arrangement of different amino acids or linking up of amino acids that give rise to different structures, and structure is explicitly or directly related to the functioning of a protein. So for instance, if a protein is arranged in a different manner from one protein, then it is only then it is able to carry different functions. So it is important that these 20 different amino acids and their random or different arrangement is what that allows these proteins to assume various structures and therefore carry on various specific cellular functions inside a body of a mammal. Or of a living being. So amino acids are the monomers that make up proteins. So you must understand that is amino acids which are. So amino acids, they have a general formula carbon, which is linked to hydrogen from one side, and then R is on other side, and then COOH, and then NH2. So this is a general formula of an amino acid which is linked to other amino acids by peptide bond. So COOH is the carboxylic acid group, hydrogen is hydrogen, and then NH2 is the amino group, and R is what is the variable chain. So this is this is a general structure of a protein. So multiple proteins are linked together by by bonds which are formed between this carbon. And it is also important to note that carbon is really the molecule or the molecule of life, because it has 4 valence electrons in its outermost shell, and it is able to bond to a multitude of different molecules, and this R is what would be different amino acids. Okay. DNA. So nucleic acids are in fact the key macromolecules in the continuity of life. So they carry genetic blueprint of a cell and carry instructions for the functioning of the cell. So nucleic acids are the building molecules, or you can say that they are what encode, these molecules encode the instructions of a living organism. So our instructions are coded within a molecule which is called DNA, and DNA are composed of nucleic acid. So deoxyribonucleic different nucleotides, so to say. Deoxyribonucleic acid and ribonucleic acid are two different molecules. DNA is a double helix molecule, and RNA is a single stranded molecule. Okay, and you do not need to understand about RNA at this point in time, but DNA, you must understand that DNA is composed of various nucleotides. So you must understand that both are made up of nucleotides. Nucleotides combine with each other to form a polynucleotide, DNA or RNA. So nucleotides are very specific in binding to one another. So for instance, adenine will only bind with thymine with 2 hydrogen bonds, and cytosine will only bind with guanine with triple hydrogen bond. So these are just for your extra information, you can read them out. But you must understand that DNA is essentially the molecule of life, because it carries the genetic blueprint of a cell, and it is composed from the monomers, nucleotides, and you do not need to understand the structure of a nucleotide at this time. Now we move on to the various tests that are explicitly mentioned in your syllabus. So this is a starch test. Sorry. For instance, you needed to check or gauge if a food molecule contains starch, then you will take that food sample. So for instance, potato in this case, peel it out, because you need to open and peel the actual fruit out, and then add iodine, few drops of iodine. You must see a blue black color. So a blue black color indicates the presence of starch. This is iodine solution, and instantly as you put the few drops of iodine solution, you must observe a blue black color in case food molecule contains starch. Then the Benedict's test, it comprises of Benedict reagent, which is copper sulfate, and then you add a small food particle to a test tube. Then you add few drops or several drops of this copper sulfate or Benedict solution, and then you put it into a warm water bath and heat it for at least 3 to 5 minutes. So a color change is observed. A brick red precipitate or brick red solution indicates a high concentration of sugar, presence of sugar. An orange color indicates a lesser concentration, yellow color even lesser, and green color a really minimal solution, minimal amount of sugar is present when green color is observed. So Biuret test is carried for proteins. Okay, so if you add protein to a food molecule, then purple color precipitate or purple rings must be observed for a food molecule to contain protein. So you can verify this. If purple color or if purple rings are formed, then a food molecule contains proteins. Okay, so these are really simple and straightforward ones, and you will be carrying them in your practical and in your lab classes as well. Fat emulsion test is for obviously fats, and ethanol is added to test tube which is containing crushed food. So you can crush a food, a bread or bread part or other flour product or other fat source, and then crush it and then put a few drops of ethanol and then shake it, and then the solution, you must take out the solution and put it into a different test tube which contains water. Now as I told you that fat would not dissolve in water because it's non-polar, and you will see emulsion. So cloudy like emulsion is observed, and that verifies the presence of fat in this food sample. So guys, this was all about this topic, and I hope you have enjoyed this lesson. These are the sources. So I forgot to mention, these are the sources which are utilized for preparing this lecture. Please let me know how I can improve my lectures, and I hope I've delivered this lecture in proper manner. But do let me know any comments or constructive criticism is always welcome. Till then, till next time, take care, and thank you very much from my side.